Hello friends, for this another day, another topic of HLA, and today we will start with the basics of HLA. I am Dr. Mohit Chaudhary. I am a senior consultant in transplant immunology and I work in New Delhi, India. This is January 2023 and you're watching channel TransConnect. So as you know, TransConnect, it has topics both from transfusion medicine and transplant immunology. So this time we are covering a very important topic, that is the basics of HLA. So I start my slide with a very exhaustive picture here. You can see that there is a lot of things, but please don't get worried. This is not to scare you at all. You can see that the central part is this MHC class 1 and you have another one, MHC class 2. Essentially, we'll be dealing with these two things, and that is why this is the main crux. So MHC and HLA are interchangeably used, and it has a wide implication. So HLA or MHC has a wide implications, right from immune regulation to inflammation to stress response to leukocyte maturation and for antigen processing. And wide variety of diseases are involved with MHC. I mean, they have a lot of disease association, many important diseases like psoriasis, myasthenia gravis, HIV, ankylosing spondylitis, abacavir hypersensitivity, that is a pharmacogenetics, and other many diseases like type 1 diabetes and rheumatoid arthritis are implicated because of the HLA genes that has been inherited by one. So you need to master this. It is a very important subject in immunity, and that is why if you know it, it has got wide implication both in transplant and in transfusion medicine. So I'll be covering these basic topics. For HLA, it will be involving the introduction, history and nomenclature, the structure, the function and the properties of HLA. So HLA or human leukocyte antigen, I would say, is a misnomer. Why it is a misnomer? Because it is not just present in the humans, it is present in other species as well. And the other thing is leukocyte. Leukocyte, obviously, it is not restricted to leukocyte or the WBCs. It is present in all nucleated cells of the body and other cells like platelets and other things. So it is not just restricted to leukocyte. So although it is a misnomer, but it is interchangeably used, and in fact it is more commonly used as an HLA when you compare it with an MHC. So MHC is the right word. It is the major histocompatibility complex, which is critical for immunological specificity, histocompatibility, and like I said, a lot of diseases, basically autoimmune diseases, are involved with it. So in human, it is present on the chromosome 6, the short arm of chromosome 6, and it covers around 4 megabase pair region, which has around 140 genes and 20% of which have functions in immunity. So very important gene which is implicated in a lot of diseases and especially for transplant and transfusion medicine. So how was it discovered? So I mean, it all started with George Snell, who started it experimenting, and he found that when a normal tissue was transplanted onto the mice, so he did a lot of experiments from mice, and he found that there was a rejection that was happening, and he labeled this system as H2, and that is where the term H came from later. So George Snell was awarded the Nobel Prize in 1980 for the same work that he did. And there were other many scientists who were working together. One of the important scientists who was also called the father of HLA system is John Dessau, who actually did a lot of experiments on French patient, and he coined the word MAC for it. So HLA later came into being, and for John Dessau he called it MAC. For George Snell it was H2. Similarly, John von Rood and Rose Payne, they were also working in a different laboratory, and they found out that pregnant women would produce some antibodies which were there in the antisera, which would agglutinate a lot of cells. So they found out that okay, there is a system which is producing these antibodies and thereby causing this agglutination reaction. Another very important scientist who actually paved the way for HLA modernization was Paul Terasaki. Why he did what he did was he developed these Terasaki trays, which are very important. In fact, we still work on these Terasaki trays for our CDC crossmatch that we perform in our lab. Now these Terasaki trays were actually a mini test tubes which he made it into a small tray, so the reactions could happen in like around 60 to 70 reactions could be interpreted and studied in just one tray, which was otherwise we had to have so many test tubes and so many reagents were used. So it is a micro lymphocytotoxicity test that he developed, because very small amount of reagent could be used because of the shape of these Terasaki trays. So he named these trays after him, and that is how we still address them. But that actually paved the way for so many research, because we had these Terasaki trays with us. So coming to the gene itself, it is like I said, it is present on the chromosome 6, small arm of chromosome 6. It is divided into three classes: class 1, 2, and 3. Class 1 and class 2 are more important for us, especially in transfusion and transplant. So class 3 is comprised of some complements and some cytokines, but unless proved otherwise, class 1 and class 2 are more important. So class 1 we have the HLA ABC, and class 2 we generally have HLA DP, DQ, and DR. So DR and DP and DQ are heterodimers, so we have both alpha and beta chains for them, which are important, or polymorphic, so we need to study them as well. So if you see, for class 1 it is HLA ABC, and for class 2 it is HLA DP, DQ, and HLA DR. So these are the class 1 and class 2 chains which are important. So what is the structure of MHC molecule? Very important. We will be studying just the HLA class 1 and class 2. So there are some basic differences between class and 1 and class 2. You see, they both have a peptide binding group, because their main function is to present the peptide to the T cells. They have to process the antigen, expose the peptide, and then present it to the T cell. So Zinkernagel actually, mastered it, and he found out there is a MHC restriction. So what happens is the class 1 molecule will always combine with CD8 plus T cells, and class 2 molecule of HLA will always combine with CD4 plus cells. So that is how he studied this MHC restriction, and he was also awarded the Nobel Prize for it. So coming to the basic differences between the two, you can see that the peptide binding groove in class 1 molecules is formed between the alpha 1 and the alpha 2. So there is alpha 1, alpha 2, and alpha 3, and you have a non-covalently bounded beta 2 microglobulin. Here, if you see in class 2, the groove is formed between alpha 1 and beta 1, right? So the basic difference in the peptide binding groove is class 1 it is between alpha 1 and alpha 2, and in class 2 it is between alpha 1 and beta 1. You can see there is a transmembrane segment which is present. In class 1 there is only one, whereas in class 2 there are two transmembrane segments. You have a non-covalently beta 2 microglobulin, and here it is combined with beta 1. So I mean, that is the main basic difference between the class 1 and HLA class 2 molecules. To summarize, where are they distribution? The another difference is where they are distributed. So HLA class 1 is generally present on all nucleated cells and platelets. So platelets also have HLA class 1. Class 2 MHC, if you see, are present on various antigen presenting cells like macrophages, dendritic cells, Langerhans cells, etc., histiocytes. So this we have already seen. They have both alpha 1, 2, 3 peptides along with a non-covalently bonded beta 2 microglobulin. Here we have alpha 1 and alpha 2 and beta 1 and beta 2. The peptide binding group in class 1 is between alpha 1 and alpha 2, and here it is alpha 1 and beta 1. Here the alpha 3 will bind with CD8, like I said, HLA class 1 binds with CD8, HLA class 2 binds with CD4, and this is called as MHC restriction, and Zinkernagel was the one who discovered it, and of course was awarded the Nobel Prize for it. The function or the antigen processed by class 1 is generally the endogenous protein, like the viruses, and here class 2 generally it is exogenous protein. We have also seen that there is only one transmembrane tail. Here in class 2 there are two transmembrane tails. Coming to another very important aspect of HLA, it is the nomenclature. So it is very important that we write the HLA, or report the HLA in the right nomenclature. So whenever you start doing it, remember it like a country code. So if you see, you can denote HLA as a country, and once you go deep down, this can be the gene. A gene can be any state. So you compare it to a state. So A may be a state, and then you have a separator. Okay, that I'll tell you later what separator means. you have two digits next to digits which corresponds to the serological antigen. That means if you start doing the molecular test, you put the separator and the next two digit will be corresponding to the serological equivalent. So this you can denote as a city. So you are going deeper now. This next would be the subtypes in the order of discovery. Suppose you have A24 first. So A2401 would be the first subtype, A2402 would be the second subtype, A2403 would be the third subtype. So depending on the subtypes, you have reached the city. So this is the country, the state, the city, the local town, the house number, and then finally, if there is something you want to, identify the house with, whether it is a brick house or it is a modern house or something like that. So basically like a postal code you can denote it. So as deep you go, you will reach your house and you will know that this is how a nomenclature of HLA is. So HLA A24 is two digits. HLA A2402 is four digits. When we say two digits, four digits, you should know what we mean by that. HLA 240201 is 6 digit and HLA 24020102 is 8 digits. So maximum you can go up to 8 digits. Many a times you will see that there is some alphabet attached to it, which can be L, Q, N or S. So L stand for low, that is the protein expression. So it can L stands for low expression of the protein, N is null, that is the whole thing is not expressing itself. S is secreted, only the secreted, and sometimes Q, which is aberrant or questionable. So basically, this is the basic nomenclature. When you do an HLA typing, you should at least report for a kidney transplant up to two digits, that is A24, which is also the serological equivalent. When you are going for a hematopoietic stem cell transplant, you should go deeper into the resolution, and that is why you do a high resolution, which is HLA A24020102, that is up to 8 digits or whatever you can address through. Anything between the 2 digits and 8 digits is intermediate resolution. So this is low resolution A24, and when you have full 8 digit, it is high resolution. Anything in between is a medium or intermediate resolution. So that is how you address the HLA here. Coming to the properties of HLA, very important. I mean, if you know the properties of HLA, you can know how it behaves, what kind of a behavior the HLA is going to do, and where it is involved. Very important two properties is constitutive expression, that is codominant and constitutive, and en bloc transfer. Now, what does that mean? Codominant means if you have one allele from your mother and one from your father, both of them would be expressed as an individual in you. So both of them would be codominantly expressed. Constitutive means in stress, it may be upregulated or downregulated, or depending upon the body structure, whether it is an inflammation or whether there is some kind of an infection, it may be constantly expressed or depressed. That is the constitutive expression. Now, en bloc means since the alleles are A, B, C, that is class 1, we also have DP and DQ and DR. So we will get one set of allele as a haplotype, which is half a set of allele from the father and half from the mother. So when en bloc transfer means the whole set which you have received from your father is going to go as it is to the son or the daughter, and similarly the whole set from the mother is going to go en bloc, that is if it is A02, B27, C7, whole A02, B27, C7 will go as it is en bloc into the progeny. So this is called as en bloc transfer. MHC restriction we have already studied. MHC restriction means CD4 will react with MHC class 2 and CD8 will react with MHC class 1. Now these two properties which I have made it bold and marked in red are very important. HLA is hyperpolymorphic. It is the most polymorphic gene. Now what does that mean? That means there are multiple combinations of each locus and its allele. So like in A, if HLA-A, if you study, you can have in that locus A, you will have multiple combinations. You may have 01, 02, 03, 04, 05. Similarly, it can go up to 100 or 200. So multiple combinations. So what you will inherit from your father and mother would be totally different from what another person would inherit. So there are multiple combinations. Like for example, in a class, there are boys and girls. So in that class, there are only two combinations. There can be a boy or a girl, unless proved otherwise. But if you see HLA, what you will inherit from that A locus, there are so many different combinations. It is so polymorphic. You don't know which gene you will inherit from your father and similarly from your mother. So everybody is quite unique in that sense because the HLA is hyperpolymorphic. This prevents us from having infection, and also sometimes when this HLA barrier is removed, it can lead to tumorous growth. So that is how our body waves off these infection by making it so diverse and hyperpolymorphic. Now the other property of HLA is linkage disequilibrium. Now linkage disequilibrium means that although the HLA is codominantly expressed and transferred en bloc, the frequency that is observed of some specific haplotype is more than or less than the expected. So for example, like A26B803 is more widely expressed in Indian subcontinent than specifically A26 alone or B8 alone. So that means that a combination, a person who wants to find a match for himself, suppose for a transplant, whose one haplotype is A26B8C7DR3, there will be many more individuals who will be sharing such an haplotype because in that community this haplotype is more common. So this process where a haplotype or a genetic consortium is more than or less than expected is known as linkage disequilibrium, and good for us that HLA follows this. That is why many a times we find a donor within our community and specific to that community that haplotype may be more common. So HLA follow a Mendelian inheritance, like simple Mendelian inheritance, and which I'll be explaining it further. So how this is followed is expressions of a MHC allele is codominant, we already know. So the particular combination of MHC allele found on a single chromosome is known as a haplotype. So together from mother and father, we will get one haplotype each from mother and one haplotype from father. So if you see this diagram, you can see that there is a combination. Half the combination will be received from the mother side and the half the combination of the alleles will be coming from the father's side. So this is how a haplotype is reached. Now, assuming there is no recombination, four different haplotypes are possible from the offspring. So if we can say the father is AB and the mother is CD, when A denotes one haplotype and B denotes the second haplotype, C is the third haplotype and D is the fourth haplotype. You can see the combination that can be formed is AC, AD, BC or BD. So this is recombination. We can leave that, but at least you can predict that the combination can be AC, AD, BC and BD. So depending upon what haplotype it is, because it is transferred en bloc. So easier way to remember is, so if you have one sibling, that is you have one brother or sister, so there is a chance that HLA identical would be 25%. That is 1 minus 3 by 4 raised to power n. So that is 1 minus 3 by 4 raised to, if you have one sibling, it will be 1. It is 1 minus 3 by 4, which is 0.25 or 25%. So this is simple HLA inheritance, which is Mendelian inheritance. So if you have more siblings, obviously the chance of having an identical sibling improves or increases, and you may have a 44% chance if there are two siblings. If there is three siblings, it is 58% chance, and the probability can never be 100% because it is 1 minus 3 by 4 raised to power n. It can never be fully 100%. So each time a new sibling is testing, there is a 25% chance. It doesn't matter. The first two siblings may have a full match. The first three may also have full match, and then the next three siblings may not have any match. So it is possible that sometimes the first two only, if you are two siblings and you may have a complete match, whereas you are six siblings but you may not have a complete match at all. So it is every time a new sibling is tested, the sibling has only 25% chance of being a match. I have already explained that HL is hyperpolymorphic. That means there is polymorphism. There is within each gene there are multiple variants of each allele, and that is why MHC in fact is the most polymorphic gene, and you should remember that, and that is why we are able to wave off so many infections that may occur if this HLA was not polymorphic. If you see the polymorphism, you can see that A, B, C, and DR are considered the most polymorphic, and that is why it is minimal that we do a typing for at least A, B, C, and DR. Also, very polymorphic are DP and DQ. So ideal would be A, B, C, DP, DQ, DR. Some are not so polymorphic like HLA-F, HLA-G, or HLA-DM and DO. You can omit that. But if there is no cost constraint, at least ABDR, if there is ABDR is non-negotiable, but best would be ABC DQDR. So in our lab we have started with ABC DQDR, and sometimes we even go for DP. But if that is for solid organ transplant, for hematopoietic stem cell transplant there is no negotiation. We have to go for ABC DQDR and in fact DP as well. So many HLA alleles were discovered. This is the slide that was, compiled in 2008. So you can see with every year the alleles are increasing, right from class 1 allele to class 2 alleles. So you can see the alleles are increasing. Almost 2600 alleles were discovered by 2008, and every time there is a mutation or something, or there are new studies that are being done, there are more HLA alleles are being discovered, and that explains the HLA polymorphism. So you can see that many labs are now even submitting, because there is an international workshop that is conducted, and you need, if you find a new allele or a new antigen that you have discovered, you need to submit it in that workshop, and the society there is going to evaluate if it is actually a new allele, and if it is approved, then you would get a notification, and that allele will be incorporated in the number of alleles that have been already discovered. So I think this summarizes the very basic of HLA. If there is anything which you have not understood, or you want me to repeat, or you want some extra notes for the same, I'll be most happy to provide you that. I hope I have opened a new sphere or new realm where which you can study, and I'll be most happy to uh, help you progress in this field, which is a very interesting field, especially with regards to transfusion medicine and transplant immunology. Thank you so much, and please keep learning.